So today it's not, as I had originally announced, going to be a talk from Michael Budden because of problems to arrive in time in Munich. So the lecture will be next Tuesday then. Therefore, today about the prediction of protein interaction sites that we began on the whiteboard on Thursday. Again, so this lecture will be next Tuesday. Don't forget, Thursday there's no lecture because of the Dies Academicus. Predict protein-protein interactions. So just to recap, for us in the course of this lecture, protein interfaces are the ones that are binding, touching. Associations are not what we are going to consider. And we want to, in fact, predict that proteins interact. Most of the data about pairwise protein interactions come from yeast to hybrid. That, again, tap pull-down menus and the like produce more data. Mass spec produces more data. Co-expression produces more data, but they are all more testing association. If you really want to probe that two things interact, that is more yeast to hybrid, carefully done, and that in fact provides most of the data. Let's begin with this typical question of homology-based inference. Can we infer interactions? So essentially it's the same question that we had before for subset localization and everything else. We have an observation. We know that A and B bind. We have a protein A prime that is similar to A, protein B prime that is similar to B, and we ask at which level of similarity here between A prime and A, B prime and B, can we predict that A prime and B prime do interact? Again, we're looking at protein interactions. That work has been done by Sven Mika. And obviously, to some extent we can do that. If we couldn't, there would not be a point in having model organisms. Proteins interact, function through interaction. If we could not study the interaction of the human pair of proteins in mouse, as you know, most experiments for human proteins are done in some other model organism. And since function always starts with interaction, if that would not be the case, there's not much of a point for model organisms, right? How well is it actually working? So this is the H-val. You remember, the blue one here is the H is the P curve. The distance here measures the distance from the blue. And in this particular case, all the values are positive. That would be negative values here. Positive meaning that everything is sort of more similar. Just let's look at 250 residues. So we're talking about things that are more than 20-something% similar. Over 100 residues, more than 30% similar. So 10 here means 43, 53, and so forth. So high levels of sequence identity between two proteins. And I'm asking, now what I'm measuring here, let me go back into the slide here. What I'm measuring is the minimal similarity. So the HSP distance that I quote is this is x1 and this is x2, is the minimal of the two, okay? So the pair A to A prime, B to B prime, one of these two has at least that sequence similarity, the other has more. That's what I plot here, okay? This is the axis of all the others. That number is very, very low, but we have to consider one of the issues here. I take all the proteins that I know interact in worm as positives. How do I choose my negatives? Everything else. Not observed for the course of this analysis means not interacting. That obviously you know is not right. And that explains why these numbers are so low. But whatever we observe, we don't expect from the chance of we have observed those today that most likely will not really impact those curves. Right? So we do know it will impact the size, will impact the number. So the numbers here, the precise value is not really meaningful. It's very low. How low it is? If we knew the entire data set in 20 years from now, maybe the number would still be that low, and maybe would much higher. We could not say. What we can say today is that unlike in the situation of subset localization, where these numbers really estimate the truth, this is just a value. What is important here is the relative value. And in terms of the relative value, unfortunately, we don't have enough data. So you see high error bars in red. But you do see, you can convince yourself that you see some line. And that line is of the type that you expect. So there's a higher value here, and there's a lower value here. You can also sort of get the idea that if you compared it two other features, other homology-based inference, structure in green, subcellular localization in blue, enzymatic activity in red and orange. You see that these lines do seem to at least not drop as early. So structure is the highest here, drops suddenly, but somehow there seems to be more signal here than here. This one here seems to be really relatively low compared to that point. So the drop, whatever the number is here, the drop from here to here, the drop from somewhere here, those are the numbers that corresponds to 60, is here, to the number of 0, is really more than for these kind of functional aspects. That much is clear. So far what we look at is really homology-based inference. Homology-based inference assuming that essentially we have a situation where a gene in a different organism does the same thing, for a pair of genes actually does the same thing. Let's expand this model. Let's just, instead of looking between the similarity of A and A prime, bring up another one, A2 prime, another here, B2 prime, and let's construct it such that A and A prime now sit in the same organism, and A2 prime and B2 prime sit in a different organism. What would you call A prime and B prime? Any word, any descriptor that jumps at you? Exactly. So these are typically what you would call orthologs, and these are typically what you would call paralogs. The model behind being that they happened at some duplication of, or through some duplication of a gene after the speciation event. And typically you would assume that A prime is in fact another copy of A in the same kind of organism. Here in this example it's worm. And typically you assume that A prime in fact can do the same thing as A, but since you already have A, has some freedom to evolve to do something new. And typically that is assumed to be the power of the way we evolve different features. The advantage of rapid gene duplication. And while A2 prime and B2 prime, we typically assume it's just the same pair in a different organism. Say in human and in worm, we're talking about the same proteins that do have to interact to do some process. And it will be the same, that's the idea, right? So therefore we assume that if we now look at the worm versus non-worm, and now we compare that to the paralog, so worm versus worm, we expect what for the next curve? Where's the next curve going to be?
Yeah, I would say that inside of the worm it should not be so conserved.
So it should be somewhere here, right?
Yeah.
So, anybody disagrees? Okay, that's of course that's exactly what we observe. That's exactly what we expect. And that's exactly what we don't observe. Welcome to biology. What we can see, we have high error bars. In fact, the error bars are so high that the data points here I cannot even show, I don't even have. These are a variety of reasons. But what I clearly see is that despite the error bars, the signal seems to be clearly such that blue paralogs, or what you would call quote-unquote paralogs, is higher than green, what you typically would call orthologs. Well, that may be more specific. So let's look at the fly. Essentially you see the same thing. And we did that for other organisms. The trouble is, when we did that, so that's almost 10 years ago, 8 years ago. So 10 years ago we started the analysis, 8 years ago we published it. But we did not have many interactions then. So we could not do that. We did it for mouse, we did it for human, but the error bars got up and up and up. And we sliced the cake in different ways, and whatever we did gave the same signal and gave the same difference. Why is that the case? Oops. Why is that the case? Essentially, we don't know. It appears as if paralogs were more conserved interactions, or can conserve the interactions, the orthologs don't. Now there are several papers that sort of confirmed this view. Meanwhile, Predrag Radivojak published a paper relatively recently, a few years ago, in which they observed a similar reality. Paralogs conserved more for the inference of Go numbers. In some sense even more surprising. And the evolving picture is that you get an advantage from having the paralog sit around and still do the same function. If anything happens to your gene, you still have a backup. Maybe you have the backup because you have to do twice the same thing. Whatever the situation, it seems to be a signal that is common, particularly strong for protein interactions, but actually also very strong for Go numbers, as Predrag's group showed. It does mean that model organisms, it's more complicated to infer protein interactions from model organisms, but that's a reality we have to live with. And that brings us to the next issue we briefly discussed on last Thursday. If I measured the same interaction twice, would I see the same thing? So in this particular case here, we have, in fact, should have taken the other one. Forget about it. So essentially you have an interaction between protein A and B. You measure it twice in two different crystals, and you ask, are the interfaces, the contacting interfaces, the same? Here are some answers. So the different colors here are simply according to what you call A and A, or how you measure similarity, and let's forget about that. Let's just see one type of bars, and you look at whatever your eyes see best. Dark, light gray. Anyway, this end here is one particular, in the inlet is one particular way of measuring the similarity of the interface. Now the similarity of the interface, you form an interface by contacting residues. So you can ask, is it the same pairing? So is that residue 5 always interacting with that residue 6 or something like that? Or you could say, okay, residue 5 here is a lysine, and here is an isoleucine. Are they interacting? Is it always leucine and an isoleucine, or is it a hydrophobic interaction, or is it a charged interaction, or whatever? Or you could ask, is in fact residue number 5 always interacting to something, whatever that is? So those are different ways of calling an interaction conserved. Or you could look at an interaction at a sphere around the interaction site and ask, in that sphere at least, do I observe in both proteins the same type of residues? Maybe they are shifted. Maybe they are like this, but still it's the same set of residues that is involved. And we essentially, Tobias, who did the work, Tobias Hamp here, looked at all of these different measures. Those are simply two different types of measures. Let me not explain how they were evolved. Conserved is here. Not conserved is here. The same thing for this one in the outer plot here. You see that most of the signal sits here in the last bin, the most conserved bin, inside the same thing. But you also see that there is some fraction of the signal for change. The different types of interfaces are in fact not correlated, so the cases that are here different are not the same that are different here. So there's a lot of difference. Let me just show you a few examples. Here, you have in fact 6 particular, the green is the central one, and the other one is one protein binding in 6 different modes. And they are absolutely different, right? This binding here is very different from this binding, from this, and so forth and so on. Here you see the blue is really flipping the way it binds. Here we have different sides, not quite different sides, but different angles in which you bind, the different ways of binding. Here again, flip sides. Those are the protein interfaces between, or the protein interactions between the same pairs of proteins in 2 different crystals. Clearly, there is a variety. Clearly, there is change, and the change is not the type of, it depends on how I define my interface, right? This is a big difference. And there are many of these kinds of big differences. The question really is, what does it mean? Is that now giving you a new way of binding? Is that something that will be settled by the environment? Is that something, we do know that the details of the binding interface matter. And we do know that in principle the same protein pair has to bind in the same way in order to do the same thing. So this seems to be a new principle out there. What is it important for? How is it steered? How is it fixed that this solution and not that solution is taken? We would have to learn about that. This is sort of the first view at some reality that seems to be indicating more complexity than we had assumed. Initially, many years ago, I had a simple idea. I wanted to predict protein-protein interactions between two proteins. So these are 9 consecutive residues in one protein, 9 consecutive residues in another protein, and I simply wanted to take some machine learning device and predict, given those 9 residue sequences, the region of interaction. And in 1999, when I had my first method developed, and it absolutely did not work. This was clearly the worst method that I've ever developed, or the most beautiful failure that I ever had in my life. And I decided that I'm going to go onto an odyssey of reducing false positives. I decided, when I looked in detail at why things got wrong, I looked at the structure and I asked myself, why did my system predict something that obviously is not binding? And the most extreme examples I looked at, I at least convinced myself that the machine learning had learned something that made sense to me. Now when you have something that makes sense and is wrong, you're in trouble. My explanation then was, well, maybe there are many things that would interact. If you would, back to your idea of using peptide binding, if you just cut out the peptide and throw that into a soup of proteins, then it may bind. But since it has to be folded in a protein, since these folded proteins have to meet in a cell somewhere, since all of that has to be happening in a coordinated fashion at some moment in time, maybe it doesn't happen. Maybe it doesn't happen because they never get near each other. Maybe it doesn't happen because they are not open to the interaction because they're folded in a different way. Maybe they interact with other domains. And all of that I call false positives. I call the ocean of false positives, and I want to try to reduce the number of false positives. The first one here is the idea of surface residues. What interacts are the surface residues, and then I had a method that sort of predicted the core residues very well from sequence, and I decided to develop a method that sort of zooms into predicting the solvent. Assuming that about half of the residues are on the surface, you reduce the number of false positives by a factor of 4. So you keep 25%, half by half is 1/4. So essentially you throw things out that are really interaction points. But most of the things you throw out are most likely not interacting. Most of the things you throw out are really the core residues that you don't care for, at least for a method that predicts interaction sites. So that was the first method that we developed. The next method was, can we in fact look at a protein sequence and predict its interaction sites? Okay, and that is what most part of today's lecture will be about, this one here. We developed that. The next idea was to use subcellular localization. Any idea how I can use that? Yeah?
Well, proteins that aren't the same locale probably won't interact.
Yeah, exactly. They're not just, again, in the same thing that the same sense that I showed you before, they don't meet. So what I show here is for different types of subcellular localizations secreted or extracellular, cytoplasm, organelles, mitochondria, nuclear, and transmembrane. Green means that these proteins from these different organelles or localizations are observed more often than expected by chance. Red, and it's 2 standard deviations more often than expected by chance. Red, 2 standard deviations less than by chance. And you see that essentially the most important signal sits at the diagonal here, with some exceptions, and part of these exceptions are membrane. Anybody can explain that exception here, the organelle and transmembrane? In the room? That's a very circular. Is it obvious to anybody in the room? Yeah. Well, then nobody says it. So obviously the point is that most proteins in organelles are mostly sitting in the membrane. So this is in fact in some sense the same as the diagonal, because many membrane proteins are also labeled organelles, so there's double labeling here. There's another point here. No, wait a minute. There's another off-diagonal. The nuclear and cytoplasm, well, that's, I guess, just an experimental confusion. Oh, how often am I going to do that today? Okay, so now, if we use that signal, in particular, given Tanya's and Tobias' new method that sort of had 18 different states, we completely reduce the false positives. Let's just assume 1/5 of the protein is nuclear. If I cut out everything that is not nuclear, I reduce by a factor of 25 the false positives. And again, I make the mistake because many proteins I will throw out that really interact. But the ones that I throw out mostly don't interact, and what I throw out is mostly really the wrong ones. The next one would in fact be to predict proteins in substrate, DNA and other interaction that is ongoing, and some of that I will show you toward the end of the lecture. Then we worked on the prediction of domains in order to define from sequence what are the regions that are the units in some sense in our method. And then we want to put it all together and predict protein interactions. So I believe I've given all over the world for almost 10 years talks. And in those talks, I essentially evolved these slides, and I showed something about those methods, and I always came to this point. And I said, now we have all the tools available, and now all we need to do is put it all together. And you know what changed in these slides? The face and the name. The story was always the same. So it was always the same person who was supposed to now put it all together and do it, and then things got in the way. And it was never as easy as it is. So that is now, essentially we have been working on all of this for 15 years now. And it's the first time that I'm going to present you the answer. Now it really works. So it's on a major revision. So Tobias really nailed it. And at the end of the year or the beginning of next year, or earlier, I'm not entirely sure about the timing, I will show you what Tobias did. So now actually he did it, and we have it. It was a long, long way. Let's begin with the data collection again. So what we want to predict here are the interfaces between two proteins that really touch. And the first question really is, do interfaces between residues inside a protein, residues between transiently binding proteins, so that are binding sometimes and sometimes not, do they differ in terms of the biophysics? Can I distinguish with different types of interfaces? What I show here is the amino acid composition difference. So difference with respect to the background, to all residues in the database. And the database used here is Swissprot. Plus means more than expected by chance. Minus less than chance is background here. For different types of interfaces. Let's get to the simpler version of it. Same story on this axis, same amino acids, but in this particular case I have only transient protein-protein interaction red, internal green, and those are homodimers. And what I'd like you to do, assume you had this plot, describe it. Please, again, when you observe data, whenever you gather data, the first step is to observe to yourself, what do you believe is the most important meaning of this plot? And that you do for yourself, you write it down, then you do it for your supervisor, and then you do it for the public at large and write it into the paper. So what are the most outstanding observations here? And you really, what jumps at you the most? What can you tell? Again, below 0 means less than all other residues. Above 0 means higher than all the other residues. I'm asking whether that particular amino acid in one of those three different types of interfaces, homodimer, internal of a protein, protein transient protein-protein interaction, external, is less for the arginine, more for the lysine in red. Again, X's are clear? Anybody? First observation.
My observation that tryptophan and threonine are all
tyrosine.
in both in the C3 cases are not in the interface.
So the first observation is that tryptophan and tyrosine are most outstanding. So you actually have two observations here. I assume that implicitly you have two observations. Those, in fact, are the most outstanding residues, and both of those stand out negatively, and both of those stand out for all three classes negatively. It's three observations. You took three observations. Anybody any idea why tryptophan and tyrosine could be?
Because I guess they are in the hydrophobic core of the...
Yep. That,
And they should be internal, right?
yeah,
I don't understand why there are cases.
that's what she says. What would your answer to that?
Because they are both in the internal and the interface.
Yeah, they're not.
No?
So your first observation was they're both negative, right? Or whatever. Let me not. You had so many. You picked so many points at the same time. But one of these points that you picked is that they're both negative. So that contradicts your assumption.
Can it also due to the fact that they are so few amino acids of tryptophan and in Swiss word, that this is not that every
We will get back. We will get back to that point. So yes, I will get back to that point. The answer in this particular case is not. But you want to make a point?
I think they're really large because they have residues. So maybe that's why they don't interact that much.
How? I get back to that point. We get back to your point later, Sabrina, but you still haven't answered. Do you uphold your argument? Or did Verena kill your argument? Or did the plot, or the observation of the plot, or the refined view, or whatever you call that, the additional view, the perspective from sitting 2 seats further to the right and back, did that change your... Anybody else wants to help him out? Defend him or...
I have...
You attacked him.
No, I don't attack him, but I don't... I mean, when all three are negative, or there are cases where all three are positive, where
We get back
would
to
the
that.
amino acids sit, which are
so,
on the other side?
Okay,
You know,
so-
there should be a fourth.
So this is the one very, very important observation in this plot. Everything here differs from 0. So if you had a plot that showed all the bars that essentially add up to all residues, then this is the moment to find the bug in your program, right? So her question is precisely the one right on. If those numbers don't add up to 1, essentially, where are they hidden? And the answer really is, one important finding here is that all of these are pairing residues. So when you look at residues that pair to other residues, they are not your average run-of-the-mill. They are really outstanding. So they differ from all the other residues. Hence, you can in fact predict essentially which residues will bind and which residues will not. It's not the same residues. That's why they all differ from 0. And that is, again, is another, so this plot is full of a few important observations, and that is one of them. The reason why I take the time to go through it is because I want you to train simply observing important data. This is what you always do as a scientific discovery. But now back to your point. I would not uphold your point, actually. She is totally right on. You may sort of try to do these, but you then, if you made those, you would have to absolutely ignore the green ones, or we have to get back to Sabrina's point of error bars, which at this point we don't have. And we will bring that error bar point up. But if there is no issue with error bars, so if the error bars would in fact say, if the error bars were sort of tiny, and we're clearly considering this a difference, no matter what, then this is clearly also a difference from there. And that clearly means that this is not about internal interactions. Yes?
I just want to ask you whether Did you want to exclude the chain-chain interactions from protein-protein interactions?
We get back to that in a minute. Well, now you're sort of, I'm the train being derailed here. I'm trying to bottle it up, observation by observation, and you bring up too many questions that lead beyond this plot. Another important observation for me on this plot would have been that they essentially differ. The three types of bars differ. If the three types of bars differ, if you believe that homodimers and transient protein-protein interactions in fact have different biophysics, then my answer to your question is yes. Clearly I want to distinguish. So the reason of this exercise, sorry, I should have motivated that more clearly, going back to what we went through on Thursday. I want to predict that a particular residue binds to another protein. I don't know the other protein, I only know the sequence. My question is, do I train my device on all residues in the PDB? Or do I only train on the subset of those that bind to other proteins? And this plot is supposed to give me the answer to that question. And if I see this plot, then I would argue that blue and red differ. Now you could sort of try to shoot me down. You could say that is part of looking at the plot and teasing observations out of this plot. You could argue that it is true that the height of red and blue differs. But it's also true that whenever you look at these, they typically sit on the same side of the distribution, while green behaves differently. So if I were to group, then green may be clearly different, but blue and red might be more similar. That is a point you may make. And it's a debatable point. By the way, here's another point that I wanted to alert you to. It's a subtle point, but an interesting one. If you just looked at the first two residues, anything that jumps at you? Sorry, not residues, amino acids. Two positively charged residues. Two positively charged amino acids. And they behave very differently, right? Again, we still haven't answered, I still haven't answered your error ratio. But they look rather different. While if you look at the negative charges here, they look more the way you would expect it. So there are some differences between red, green, and blue. But between the negative charges, they are more similar to each other. Okay, let's keep it there. And the question, of course, is, are these differences meaningful? That brings us to, you said tryptophan and tyrosine, they are exceptional, but actually not that exceptional. So the counts of these are quite high. How can we answer whether the differences are meaningful? Well, we essentially have to look at statistical significance. And statistical significance, that is what I told you, the chi-square test showed that the statistical significance of these differences are extremely high. And that, however, was also true for things that didn't make sense. So we had to come up with a new test that is not using chi-square. And the test we did is this find self test. So we have a data set, internal homodimers, external interactions. We pick out, so these are a few tens or 100,000 points in there, and we pick out 1,000 from each pot. We pick out another one from this one here and ask, these 1,000, what are they most similar to? If they are most similar to that, it finds itself, and on the set of 1,000 at least, we clearly established that there is something that makes them differ from each other. And the way we compare that, the JS is the Jensen channel information here, that's simply the way we measure similarity between the data sets of 1,000 picked out balls, amino acids. And then we repeat this experience 1,000 times, and we see this. Now, 6 different types of interfaces. Let's just go through what they mean. Internal, domain, clear? Now we have homodimers, and I have heterodimers. Now, we distinguish here, following an annotation that Janet Thornton introduced at some point, the homo-obligomer and the homo-oligomer. What we mean here is this is a homodimer that has never been observed. So the interface between these homodimer is such that I've never seen this stretch to observe, to interact with anything else. So that's why we call it obligomer, or that comes from Janet Thornton. It's obligatory to be together in some sense. While in this particular case, it has a choice. So the homodimer could be together, but sometimes it's not. Okay? That's a homo-oligomer. The same is true here for the hetero-obligomer and oligomer. The hetero-obligomer in this particular case is simply, we are looking at two different chains of the same protein, and we call them obligomer because they are co-expressed. They come off the ribosome, they are one protein. They cannot escape from each other. They may escape from the interaction, but they are stuck, right? It's one long protein. While in the case of the hetero-oligomer that we talk about here, that's really the transient external protein interactions. So we're talking about two proteins that in some point in time come together. That is what we typically call a protein-protein interaction, a transient protein-protein interaction. So that's the case that we care about. And what you clearly see is that when we do that 1,000 times, the diagonal is the highest point, right? And now actually back to your homodimer story. They also, the blue ones here, also clearly stand out. They stand out from the internal interaction and they stand out from the external interaction. They clearly stand out, in fact, even from the hetero-obligomer, so the interaction between two residues in the same protein that are different regions of the same thing. When you look at the confusion, you would, for instance, jump here. The largest confusion is this one here, which is domain from the hetero-obligomer. So that means the residues that are interacting between two domains in a protein and two different chains or different units. And that's fairly trivial because essentially it's the same thing, right? It's the same biophysics. It's just by one definition you call it the domain, by another definition you call it a chain. Ultimately, that is a completely understandable confusion in the sense that's actually sort of almost the same. The next highest confusion here is to internal. That may be an important signal, but mind you, this is only 6% of the signal. But that may be an important signal. It may also be a mistake in labeling the domain. So the degree to which the residues and domains are the same differs between different assignment methods by more than 10%. So that might be just that. Overall, however, you see that the part that we care for here, transient protein interactions, really stands out. In some sense, 90% unique. And that you can now use to develop a prediction method. So far what I had is I picked out 1,000 points at a time. So that is not really a prediction method. If I want to put that into a prediction method, I really have to build my data set. I take PDB. This is outdated. Now we have over 100,000. I extract the interaction. You look at the sequence unique subset of the PDB, and you know why we need to look at sequence unique subsets. Once I have a sequence unique subset, the question is what criterion for similarity we take. You said that already. Here's a particular choice about the closest atom is 6.5 angstrom. There are slightly different ones which I can use. Then we have in fact the PDB chains. This is assignment. Now let's not get into that. We have a method. At the end of the day, we have a data set. Now we can apply machine learning. In order to apply machine learning, again, what we want to predict is a particular residue is involved in protein interaction and external protein interaction, and you still have to make the point that there's a consecutiveness of a signal. That's here. So when I look at a stretch of 9 consecutive residues, I ask how many of these 9 bind in a protein interface. If I define my protein interface by closest atom is 6 angstrom, blue, or closest atom is 4 angstrom, yellow. In both cases, yellow, the distribution is more on that side. Blue, the distribution is more on that side. Either of them is such that clearly it's not 1. So every interface of 9 consecutive residues that is somehow in an interaction has typically more than 1 residue involved in an interaction. In fact, the average for 6 angstrom is here, and 6 ballpark, the other one for 4, but in fact half of the window. Whenever you cut out a window of 9, or you have a window of 9 that involves at least 1 residue in the interaction, you typically have 4 or something. That means you clearly have a sequence consecutive signal, and you clearly have hope to do machine learning for this. Now, how do you choose your input features? I want to see you sleep. Not good. Too much attention. Any idea what kind of information is important? Yeah? Was that?
The secondary structure maybe.
Why would that be important?
Yeah, because if you have an alpha helix and you would say that for the outside of the helix there is an interaction.
Can you name any protein that would do something like helical bindings?
Colicos.
Yeah, exactly. So we know immediately, we have immediately some suspicion of some domains that we know interact, bring together helices. We clearly have a, you all have seen coil interactions, I assume. So one way in which they do is they bring together 2 proteins and actually even entire cells. So these coils winding up have an immense power to bring about proximity. And yes, any other thing that you have seen before? Leucine zipper is another reason why that is called the zipper, because they zip up for leucines that sit on the face of the helix that bind to each other. Beta strands. There are, in fact, examples of proteins. Maybe you have seen some where you really the mad cow disease is an example. Essentially, is aggregation. So that is essentially for some of your last semester's projects, right? Where you, through aggregation, you get binding, and this binding, typically we think about it as a disease state. But this is just a way in which beta strands aggregate. The Alzheimer protein is an example for that, where we have aggregation for beta strands that sit on the surface and essentially are very gluey. Another example. So yes, secondary structure would be an important feature. Anything else? Surface, right? We already discussed it. What will interact in the protein is clearly the surface. So you can predict surface. Yeah?
Also probably hyperphobicity.
Absolutely. And we clearly, again, in the signal here, saw for the residue composition, we saw some signal. In fact, that was the one you jumped at. Maybe your jump was not, that part of the jump was not supported, but clearly there was a hydrophobicity signal in there. Anything else? Conservation, of course, is another one that will help. And then we will just have the, so we will begin to in fact use the sequence, do an alignment, filter out the related ones, run a bunch of methods that predict solvent accessibility, secondary structure, that look at globularity, predict domains, membrane helices, predict the subset localization, put all of that together, and then you get a method that essentially uses profile, predicted 1D structure, some predicted aspects of function, and then you do some cross-validation here, make sure that you really have a data set split that is appropriate, you have cross-validation, and you get this. This particular case actually is only sequence. Correctly predicted by observed, correctly predicted by predicted. That's the light blue here is the signal when you use only sequence. Does it work? I do understand that I could have made the blue a little bit more visible. Please don't let that influence your statement. So here is blue, for those of you who have eyes like I. So there's something there. In order to know whether it works or not, what do I have to do? By the way, I scale in my prediction system through this line here by simply looking at the strength of the prediction. So the ones up here are the stronger ones, so where the difference here, both of these cases I predict that unit, which is the binding, the transient binding. In this particular case the prediction is stronger, so those are the ones on that side. Okay, so what do I have to compare it to in order to see whether it works at all? I need a random background. That is the random background. The difference between these two plots here is simply I scale up here to, in fact you see this is 0.03%, so it's a tiny subfraction that I have, and that's an important thing, and that may be why Dennis says that it doesn't work. But this is random. There's some fit here that considers the error estimates. So, does it work? Dennis, do you still say it doesn't work?
I'm not really sure.
Oh, come off it. You're not sure that orange differs from blue?
No, I'm sure that it differs.
Oh, good. So what is bothering you?
I don't really get the coverage of it observed.
You mean what it means?
Right.
Essentially this means, so I have a bunch of residues that I know are binding transiently. And I look at only the very, very, very strong predictions. And in fact this is this is a 1/10 of a mil per mil. Okay? That's where we are sitting here. It's a tiny fraction. In fact, what I can tell you is that the fraction we are at here, about this, somewhere this number here, I believe on the next slide,
So
it says that this one here is a number at which I have 1 in a protein. I predict an interaction site in every single protein on average. Some proteins not and some more, but on average, in every protein in my data set I predict 1 residue at this level, at 94%. Or whatever the number is here. Okay? That's what you would get randomly. So, can I do something?
That looks like there may be a lot of false positives, right?
Yeah, I make many mistakes. In fact, you know, as you saw here, that is the end of the curve. I can just not dial through the curve much more. That's the end of what I can reach. So, one way of seeing that is essentially if you move further, you hit random. So after this, again, it's a tiny, tiny, tiny fraction here of the curve. After that, you hit random. Okay? And that is a small subset, yes. But for that small subset, I'm absolutely above random. And in fact, if I looked at some of these cases, and that's what you should clearly do next, in a situation like that. So first of all, in terms of perspective, what does it mean? So it's 1 per protein, that is already important. And then you look at some of these cases, and actually it's surprising how informative all of these cases are. So the system clearly picked up a signal. And a signal that undoubtedly is interesting. It's unclear whether that now is something that you can use. I want to predict that protein A and B bind, and this is supposed to be a filter. How much that helps me is unclear. But I have something, and the effect that I can do that only from sequence is surprising. I can use this one here to filter and improve the signal a little bit, and then I can put in the secondary structure, solvent accessibility, hydrophobicity, the profile, and I get to this. So that's the best method. Now now I'm dialing through the curve, and I'm clearly always better than random, and I clearly pick up a signal that is much better than random, and obviously very interesting. Let's just look at the issue of hotspots. Let's look at these very strongly. There seem to be some for which I predict very strongly correct. So for the sequence only signal, those were the only ones. By using solvent accessibility, secondary structure predictions, high publicity, and alignment information, there are many more that I can predict even at higher level of accuracy. Are they outstanding? So let's get back to this concept of hotspots. Anybody in the room has an idea what the concept of hotspots is? What's the idea? Have you heard of hotspots? So we're not talking about your electric plate and finger and ouch. This is more the idea that when you have an interaction, so in an interaction between two proteins, you may sometimes bury as many as 30 residues or 20 to 30 residues. Very often, half of all the delta G of binding is carried by a single interaction. So you typically, for every interaction, there is one residue that you change, and you lose half of the delta G of binding. That is what typically is referred to as a hotspot. So the feeling that if you is the same zip curve that I glossed over before, where you see what the very many residues contribute very little to binding, and some residues contribute very much to binding, and that got people into the world of hotspots. People who don't believe in hotspots say, well, actually, there's nothing special about them. Just they're a little bit stronger. But there's a scale, like everything in chemistry. So it's not true the idea that some things are outstanding. So the concept of hotspot is one that is not meaningful to understand interactions. And there has been a debate between drug designers on this very issue for a long time. Now, one way of getting at hotspots is essentially residues that are essential for the interaction is more often that you knock out one particular residue, you knock out the interaction if it's a hotspot residue. And that's one way of testing hotspots if the interaction knocks it out. So we can now use our system and simply look at the very strong ones and see how they in fact predict hotspots. Here's an example. This is the binding between the V1 domain of CD4 bound to the glycoprotein in HIV, 120. And the example was chosen because when Jan and I worked on that, his friend had done the experiments on this one. Red are, I can't remember how many mutations they did, but a large set. And red are the ones that had a strong effect on the binding. Oh. This is predicted. So this is just put on the image. We don't know, we start from sequence only. The sequence only view, this purple is what we predict. This is just shown on the structure. Shown on the structure, you see sometimes there's too much purple, so this is not observed here, this particular residue, but it's extending, it's essentially bridging the two observed ones, and sometimes it's missing some. Although I don't see that at the moment. Yes, here. This bridge here is observed, but it's not predicted. This back over here is not predicted. But overall, this is sequence-only prediction. It's rather impressive. This is one particular example. So the particular example brings up the issue, is that enough to publish? No, you need a larger data set. Here's a larger data set on data that we in fact had not seen before, alanine scanning data of hotspots. And now this is not protein interaction sites, this is really hotspots. The system is the same as before, has not been retrained. So hotspots defined as in the alanine mutating that residue alanine knocks out the interaction. And you see that some 10% of all of these cases we predict without making a mistake. 60% we predict at 60% accuracy. Which is remarkable for a data set that has nothing to do with what you try to do in a machine learning device. But here's another aspect of that computational biology approach, which is, I told you there's a debate whether hotspots exist or not. This is one particular way of doing the experiment and getting the data. Now I'm saying that our network or machine learning device learned about hotspots, although it was never put in. We never had the concept of hotspots. All right? We never had this idea that anything is more meaningful. We take an experimental data and we can predict something. This only works if there is something in the data. Okay? This is like data mining. This is now where we in fact take data, predict something, get a clear signal. That clear signal proves that some other experiment is right and that some other concept, in this particular case hotspot, is meaningful. Right? Which is an amazing moment when you sort of can, in some sense, settle a debate between experimentalists with a computational device that doesn't fit the data. It has nothing to do with the data. How can the device learn about hotspots? Essentially, the point is that the only thing in your data that is consistent are really the strong interactions. And that's the way it has zoomed in it. By doing the machine learning right, you zoomed into the part that is consistent. And most consistent is the strongest signal. As simple as that. The most important part is evolutionary information, but the predicted 1D structure is also important. That simply sort of shows how much of the hotspots you can get. Some of that by hydrophobicity, some hydrophobic moment alone, some by sequence only, some by evolution alone, and this is evolution plus sequence plus predicted structure, and this is other aspects here. So that's the full signal. Again, now we can, the idea that we had then is, since we can now predict the signal, the site, so you can look at the sequence and predict the part that would involve binding to other proteins, we can find motifs and go out and find similar binding interfaces. And in some sense we have tried that, and we received we got some signal, but we never published. Now the other issue that we can ask is when we look at network features. So there are two different types of proteins. Some proteins bind to many other proteins, and some proteins don't bind to many other proteins. So when we look at how many protein interactions we observe in a particular protein, maybe from that I can predict whether I'm going to have a protein that binds to many others, right? So I have a device that predicts from sequence alone how many residues are involved in binding. My argument is, if I just plot for every protein how many I predict, then the ones that have many interactions I've predicted should be proteins that have more binding partners. Are you happy with that logic? You're never happy with me. It's somebody else. Sorry. Yeah, you know, I'm beginning to be too predictable. So there's a flaw in the logic. That's true. But what is the flaw? Again, I'm going to plot. So if you simply plot the number of interaction sites, so the number of residues here that you predict by this method to be involved in external interactions, versus the number of protein binding partners a protein has. Don't you expect some sort of, okay, reality is almost more complicated. But in a simplified universe, wouldn't you expect some sort of linear thing? More interaction sites, more interaction partners? Why not? Okay, it may go into steps here. More happy now? Or there's something substantially wrong in this view. It's not the little ones here.
Some proteins bind to the same interface of another protein.
That is essentially what is wrong. Exactly. That's the distinction between a date and a party hub. And wait a minute. Oops. So that's what we expect. And that is in fact Marc Vidal. In this paper here, Marc Vidal, who is one of the people who pushed these two hybrid systems the most, is Harvard Met. And in this paper he introduced this notation between date and party. So the date hub is the hub. Hub is a protein that binds many other proteins, and the date hub is the one that has the same interaction sites and binds with the same interaction site many others. It's like you're going out on a date, right? You meet one person, and then you move on, and you meet the next person, but you look with the same face at that person. The party hub is the idea you're standing at a party, and you're connecting to many people at the same time. So you're talking to the person in the back of you, you're talking to the person left and right. So essentially you have many interface sites, and you interact with many other people in the party with different, but they have a different distance to you. They're not one-on-one. Different binding sites. You could also say that the date hub essentially is something that is at different time points, while party hubs are hubs that interact at the same time point with many others. Okay? And this clearly is something that will confuse our image, because what we essentially predict here is many interaction sites. So if we saw a correlation, we would expect the correlation only with a party hub. And that's what we see. So the party hub line is going up somehow with the number of predicted interactions, while everything, non-hubs, and the date hubs stays down. So this is in some sense, the signal here is less fancy. Plot, just a 2D plot, you still see the same thing. Now the gray is behind it here, non-hubs is gray behind. Party goes up, date not. Okay, the signal is not that strong, but it's still remarkable because you predict something from sequence only. All we needed to do here is to start with the sequence, and we predicted something, and from that something we could see some correlation. The flip side of the coin is now what makes you become a date hub, and that is part of what I didn't really get to last semester. Just wanted to make this observation to close that story. And that essentially is unstructured regions. Long floppy loops. Long floppy loops you see much more in date hubs. So when, in fact, since you keep repeating this IDP word, so there has been an observation that IDPs, so in non-regular regions, disordered regions, or whatever you call them, are more observed in hubs. That has been a publication that was around for a while. It's not true. What, however, is true is that they are more prevalent. Some particular type of disorder, in fact, the loopy type, it's only the loopy type, is prevalent, very much more observed in date hubs. That's one system by which you can imagine, and those kind of plots, there are many of them around, where you really see that the unstructured one sort of adopts to different binding sites, while many different binding points is like a transcription factor, allows you to bind to many things at the same time. With that, I successfully jumped through 130 slides. And we are at the end for today. Next Thursday, again, is no lecture because of the Dies Academicos. Next Tuesday, we will, Michael Budden will talk about Bayesian networks for the prediction of subset localization. Thank you for your attention.